Hi, my name is Krishna Patel. I'm a senior at Oglethorpe University, and today I'm going to be talking to you about the role of glial inflammation on traumatic brain injury recovery. Is it protective or problematic? So, what got me interested in researching traumatic brain injuries is my experience working at CHOA, the Children's Hospital of Atlanta. Um, I've worked with patients who've suffered from traumatic brain injuries that suddenly cause a change in their personality or cause them to lose motor control or speech. And this change, as you can imagine, can be very devastating for a child who is one day functioning normally and the next day loses all ability to control their movements or to speak, communicate with others. And this change can be either immediate or it can manifest after years. So there is a lot of mystery in this field that still needs to be discovered. So it is a growing area of research. So the primary injury site of most traumatic brain injuries would be the cortex, which is the most superficial part shown in part A of this diagram here. And the cortex is connected through a circuit or neuronal network called the corticothalamo-cortical loop, or the corticothalamo-cortical circuit. And it's connected to the thalamus, which is shown in part B of this diagram here. And the thalamus can suffer from secondary damage, so although it doesn't face the brunt of the physical impact of a traumatic brain injury, it does still get damaged in terms of the functions that the thalamus is responsible for. So the damage to this neural network can result in deficits in sensory processing. It can result in disturbances to sleep, and it can result in seizures, which are shown on ECGs as epileptic spikes. So the cortex and the thalamus are involved anatomically in the corticothalamo-cortical circuit or loop. On the molecular level, the experimenters of this first experiment I'm going to be telling you about looked at a molecular pathway called the complement pathway, and the C complement pathway. So the C complement pathway is implicated in inflammation, and it can also lead to neurodegeneration and the degradation of synapses when it's activated in a disease. Normally, it's also involved in the development or the formation of normal synapses. So the experimenters in this study hypothesized that by increasing the expression of this molecule C1q, which is the initiator molecule in the C complement pathway, this would increase the long-term inflammation and the neuronal loss that's seen after a mild traumatic brain injury. The second part of the experiment, the experimenters manipulated C1q by producing an antibody that would block the expression of C1q downstream in its pathway because it's an initiator molecule. So the experimenters hypothesized that by blocking C1q, this would decrease the neuronal loss seen after traumatic brain injury, as well as the inflammation caused by the traumatic brain injury. They also hypothesized that blocking C1q would reduce the number of epileptic spikes, which are indicators of seizure activity, and it would also reduce the loss of sleep spindles. So Fewer sleep spindles are associated with pathologies like sleeping disorders and epilepsy. So in the setup for this experiment, the experimenters subjected mice to a cortical impact by hitting them with a metal piston to produce a 3 mm dent in the skull of these mice. After these mice were hit with a metal piston, they were left alone for 3 weeks, which is the latent period. During this period of time, the brain undergoes maladaptive changes and adaptations following any brain injury. So after the latent period, the mice were then split into two groups. One group of mice was administered the anti-CQ1 antibody, and the other group of mice were monitored for epileptic activity in sleep spindles. So briefly, the mice that were administered the antibody were injected with this antibody 2 times a week for 3 weeks in their peritoneum, which is the sheath covering organs in the abdominal cavity of the mice. After they were administered this antibody, these mice were euthanized and their brain tissue was dissected. This tissue was stained to fluorescently tag these protein markers. C1q is a protein marker for the initiator molecule in the C complement pathway. GFAP is a protein marker for astrocytes, which are glial cells involved in maintaining the blood-brain barrier and maintaining the homeostasis of the central nervous system. So excess GFAP is also implicated in after brain injuries because astrocytes will produce a lot of GFAP when they're repairing damaged brain tissue. Mu-N is a protein marker for neurons. So this protein marker can indicate neuron loss if we see less fluorescence for mu-N. So after this tissue was stained for these protein markers, it was examined under a microscope. So I'm going to be talking about two specific proteins that were fluorescently tagged in this experiment. These include C1q and mu-N. So according to this diagram, you can see that in the cortex, there is a lot of fluorescence in the ipsilateral side to the injury, which is here. That just means that it was on the same side of the brain injury. And it's pretty bright here that fluorescence reduces as we look specifically at the thalamus and the reticular thalamic nuclei, which is the area outlined with the dashed lines here. There's still some fluorescence, but notably less than the cortex, which makes sense because the cortex is the primary site of injury, and this is a secondary site of damage. A close-up of the thalamic reticular nucleus shows some fluorescence here, indicated by the bright red. So the reason why this is compared to the contralateral side is because this is showing you, again, the tissue on the same side of where the injury occurred, and the tissue on the other side of the brain where there wasn't any cortical impact will show or should show a control where you don't see fluorescence, and that's what we see here in the cortex, thalamus, and the reticular thalamic nuclei. We don't see any expression of C1q. Here, when we look at the diagram for mu-N, On the ipsilateral or same side of the injury, we see very bright glowing region, the primary site in the cortex. As zooming in on the thalamus, we can see some fluorescence here, notably less than the cortex, which again makes sense. And then here in the reticular thalamic nuclei, you can actually see, especially in comparison to the other side of the brain where there wasn't the cortical impact and the injury, there's less fluorescence. And because the new N is a marker for neurons, this shows you that there was neuronal loss after the traumatic brain injury on the same side of where the injury took place. And this is just showing you the effect of a traumatic brain injury before any manipulations. Okay, so this diagram is showing you the effects of the C1q antibody, which blocked the expression of the C1q or initiator molecule of the complement pathway. So in the reticular thalamic nucleus, you can see that there's a lot of different colors going on here. So to break it down to the most important points, I want you to look at the area in blue, which is going to show you the part or the C1q expression in the brain after the mild traumatic brain injury and after the administration of the antibody. And then you're going to see also the sham, dark gray is the sham or the model of the mice group that wasn't subjected to the cortical impact or the traumatic brain injury. So these mice were not hit with the metal piston. And the sham plus the vehicle shows the mice that were not hit with the piston and also not administered any antibodies. So looking at those two groups, you can see that there is an increase in the fluorescence of C1q or the expression of C1q, which shows you that because C1q is implicated in inflammation, there's likely the presence of inflammation after the traumatic brain injury. Also, looking at the red here in the same figure, which is is showing the mild traumatic brain injury without the administration of the antibody. So in mice that weren't given the antibody but were hit with the piston, you can see that they expressed C1q at a higher rate than those that were administered the antibody. And this makes sense if seeing that the antibody does work to reduce C1q expression. And in this figure here, we can see that the fluorescence of mu N is increased after the mild traumatic brain injury and with the administration of the antibody, showing that there's more neurons because mu N is a marker for neurons. In the red, which shows you the mild traumatic brain injury without the administration of the antibody, you can see that there is a markedly lower expression of mu N, which means that there were less neurons. So this indicates neuronal loss, especially because when you see in the sham mice before the cortical impact or without a cortical impact, their expression of mu N is much higher, above 1. So in this experimental study, the experimenters manipulated the C1q molecule, which is the first molecule involved in the C complement pathway, a pathway involved in neuroinflammation. And the experimenters found that by blocking C1q expression, by producing an antibody for C1q, they reduced inflammation and neuron loss following a brain injury. And in the last figure, we saw an example of this where the expression of mu-n was increased in mice that were subjected to the mild traumatic brain injury and administered the antibody, as opposed to a lower expression of mu-n or neurons in mice that were just subjected to the mild traumatic brain injury were not given an antibody. The experimenters also found in the study that the C1q antibody prevents epileptic spikes, which are spikes that indicate seizure activity, and it also reduces the loss of sleep spindles. So we see fewer sleep spindles in patients suffering from seizures. On this slide, you can see an example of 7 different types of epileptic spikes shown under the diagram for epileptiform discharges. And you can kind of see how those spikes are erratic. Below this diagram, you can also see an example of sleep spindles, which are concentrated spikes seen here. Sleep spindles are depolarizations that occur during non-REM sleep. And again, this sleep is really important in memory consolidation. So when you see fewer sleep spindles, this can be a sign of seizures and of sleep disturbances. So in this next experiment, the experimenters injected pigs with an LY protein, which is a dye that's usually impermeable to cells, so it won't cross the plasma membrane into neurons. However, when neurons are damaged, the LY protein does permeate through the plasma membrane. So this protein was injected into the brain ventricles of these pigs, and then these pigs were subjected to closed head rotational injuries. So the majority of traumatic brain injuries are actually closed head injuries, and this experiment was intended to model the majority of these cases that are underrepresented in clinical trials. So the pigs that were subjected to these rotational injuries were attached to a padded bite plate and mounted onto a Hyge device. And they were subjected to one of 2 different types or levels of the rotational injuries. For mild traumatic brain injury simulation, they were subjected to a coronal rotation in which their head was turned this way, as you can see in the first part of step 2. And for the severe injury simulation, their head was rotated up and down vertically in such a direction like this. And so after these rotational injuries made their first round, these pigs were subjected to repeated injuries every 15 minutes. After the last injury, 15 minutes, the pigs were left alone for 15 minutes, and then they were euthanized and their brain tissue was dissected. And the protein markers for LY were stained with a fluorescent tag, and this tissue was examined under a microscope. So for this experiment, the scientists involved in this study hypothesized that the density of microglia which are glial cells involved in the immune response of the central nervous system. The scientists hypothesized that the density of these microglia would increase in regions that were proximal or closest to the site of injury or damage. They also predicted that the shape or morphology of the microglia in this area would be more reactive as opposed to being typical of the normal or ramified microglial type. So in this figure here, we can visualize that A shows a ramified microglial cell, and ramified is another word for branch. So we see that the normal or branch microglia extends its branches quite far, and this is in contrast to panel B showing a reactive microglia, which is much more compact. This makes sense with the experimenters' hypothesis that in areas close to injury, we should also see a higher density of microglia, because if microglia are more reactive after neurons are damaged, then they should also be able to pack more densely, because the reactive morphology of the microglia, as you can see here, is much more small and easily packed. So in this diagram, we're looking at the density of microglial cells, proximal, medial distance, and distal to sites of neuronal damage after the rotational brain injuries. So in the mild impact model where we had those coronal rotation injuries, we can see that in mice that had the absence of the LY protein, there is only about 100 cells per square millimeter. Whereas in the cells that are more proximal or closer to the site of neuronal damage, we see a much higher density at 400 cells per square millimeter. Again, the cells that have LY protein absent, this indicates that the LY dye did not permeate through the plasma membrane of these neurons, and usually because LY dye is impermeable, this means that those cells that did not have that permeation were not injured, and thus they were the plasma membrane integrity was not compromised. So in the severe impact model, we also see about the same here in cells without the LY dye permeation, about 100 microglial cells per square millimeter, but in cells that are closest to the site of neuronal damage, in the severe impact model, we see about 300 microglial cells per square millimeter. So to summarize the big takeaways from both of these experiments, for the first experiment, the scientists involved in this study showed that the C1q antibody did reduce inflammation and also reduce the loss of neurons following a brain injury. And we saw that by the increase in mu-N expression in mice that were subjected to the traumatic brain injury and administered the antibody, as opposed to the mice that were just subjected to the brain injury and had that empty vehicle treatment. These experimenters also showed that the C1q antibody reduced at the epileptic spikes, and it also led to the loss of, or led to a reduction in the loss of sleep cycles. In the second experiment that we just went over, these experimenters showed that the activation of microglia or reactive microglia were located in patches most proximal or closest to damaged neurons. Damaged neurons were indicated by the presence of the LY protein, which is usually impermeable to the plasma membrane of neurons, but in damaged neurons, the plasma membrane integrity is compromised, and thus the dye can permeate through the membrane. So in these cells that are damaged, we should see LY protein present. In the cells that were positive for LY protein, we saw higher microglial density, and this supported the hypothesis that the damaged proteins positive for LY will have a higher density of microglia. Microglial cells are also the immune cells of the central nervous system. In addition, the experimenters determined that the microglia have a more reactive morphology in areas that are closer to these damaged neurons, and reactive microglia are more compact, thus allowing for denser packing. So in these experiments, we see that although inflammation is generally a protective response embedded into our immune system, these experiments both show examples of inflammatory or pro-inflammatory markers that cause more harm than the